Hello, and thank you for inviting me to talk here. I think, as Albert said, I'm not actually working on livestock, so this is going to be a bit different. And I think the reason I was asked to give this talk is because the thought was that even though this is all human, it might be something that's of interest in livestock, because there you can actually control admixture much more than in humans, where it's just a coincidental thing that kind of helped us answer a question. So methylation in human is quite interesting, because it is not only affected by the kind of cis genotype, but also by the environment. And it got people relatively excited, because the thought was that it could be a kind of more global measure in later life of your kind of lifetime environmental exposures, which are very hard to control and measure in humans. And also, if methylation were to be causal for diseases, and we know that there are many methylation differences associated with diseases, then if it can be affected by environmental factors, that's obviously great, because we have a direct intervention pathway. And so when people kind of started measuring methylation in humans, one of the first things they did is they started comparing it between populations. And the kind of upshot was that at around 10% of methylation sites, which were on the methylation arrays, you saw differences between populations in methylation. But the kind of obvious question is that a difference in genetics between the populations, or is it a difference between the kind of gross environment between populations? And because these are humans, we can't just take humans and move them into another environment and do a kind of experiment. So there didn't seem to be an obvious way of kind of disentangling what is actually driving these differences. And so when we looked at this problem, we kind of thought of a kind of natural experiment that allows us to disentangle these differences. And that natural experiment is an admixed population. So in an admixed population, the genotype of each person is a kind of, patchwork of the kind of haplotypes inherited from the two ancestral populations. And because each person has a kind of different patchworks, if you look at individual methylation sites, then across the genome, locally, each methylation site will be in a context of a specific genotype from a specific ancestral population. And as you go along the genome, this kind of varies. So if you look at the first methylation sites, the kind of genotypes of the first two people are in the kind of blue ancestral population context, and of the third is in the green ancestral population context. And the next methylation site, it kind of changes, and you've got only the first person in the blue ancestral context and the bottom two people in the green ancestral context. And that's kind of ideal for us, because we have the two ancestral genotypes in the same environment. So we've kind of broken the confounding of genotype and environment by keeping the environment in a sense constant. And even better, if when you go along the genome, because you could kind of assume that, okay, so maybe there's still some residual correlation between, people who are kind of have more ancestry from one end or more from the other. But as you're going along the genome, actually the environment is entirely randomized with respect to the ancestral genotype, because which bit of the genome is specifically inherited from which ancestral population is entirely random for each individual person. So the only thing we really need is knowing which bit of the genome in each person comes from which ancestral population. And there are various methods to do that. We used a method called RFMix, and I've just stolen the figure here to explain how it roughly works and what you need to get this working. So what you need is you need genotypes from the ancestral populations, which you kind of assume are pure ancestral population. And you give them, or you take them as a kind of reference panel, and then you look at small windows of the genome. And you essentially train a classifier to classify whether any new kind of haplotype in this window comes from one ancestral population or the other ancestral population. You then run that along the chromosome of a new individual. You get a prediction for each window, and then you, in a sense, phase the prediction. So you go along basically with the forward-backward algorithm to infer kind of continuous bits coming from one ancestral population to another. And that works relatively successfully. So once you've done that, what you can do is you look at a specific methylation site. You take a window around the methylation site, and you essentially run an EWAS instead of on genotypes on the kind of fraction of ancestral population in that window. Obviously, as the window size changes, that changes. You know, if you look at a very small window, then you can imagine this is essentially like an ancestral genotype. So, you know, because you've got 2 chromosomes, you'll have kind of 3 different values. You'll have either 0% of one population, or 50% of both population, or 100% or 0% of the other population. But as you make the window bigger, you kind of, like if you extend them to say chromosome lengths, your proportions start resembling the overall admixture proportions of the population. And this is essentially what we did. So as our study population, we took a population of patients from Colombia, and we measured methylation in both whole blood and kind of colorectal tissue, healthy colorectal tissue, and we genotyped these people. And the Colombian population is a very good kind of model here, because the South American populations are in general an admixture due to rather obvious historical reasons between the Native American populations, which used to live there, and then the Europeans who came in, and then African populations which were brought in due to slavery. So that was our study population. When we looked at, so what I just described, this admixture, so these are the kind of global admixture proportions. So in the left 3 columns, you see reference panel for essentially Spanish people, who were the people who mostly colonized South America. Then kind of African populations and Native American populations, although you can see that the Native Americans are already a bit contaminated with European kind of ancestries, because it's very hard to find essentially pure Native American genotypes these days. And the last block here is our study population, where you can see it's largely an admixture of Native American and the kind of European, with a smaller fraction in green of African ancestry. But these are the kind of global admixture proportions for each individual. We can do that better. So if we take, where's my point? Oh, sorry. If we take the kind of genome of an individual, which is this patchwork from the 3 populations, we can disassemble it into kind of 3 virtual genomes of kind of pure ancestry by masking out everything that doesn't belong to the ancestry. And if we then do kind of not quite standard PCA, but PCA accounting for the fact that we've got a lot of missing data in there, we see the kind of picture that we would like, that the kind of virtual partial ancestral genomes co-cluster with kind of reference genomes of the populations, which are the kind of broad ranges of the ancestral populations are indicated by these outlines. And the individual dots are the kind of virtual ancestral genomes, and the gray dots are the full admixed individuals. So we're quite confident that we are kind of pulling out individual regions which represent the kind of ancestral genetic context. And then we run our EWAs, as described previously, on these kind of local proportions of ancestry. And we do find quite a lot of kind of DNA methylation sites across the whole genome, which do show kind of differential methylation based on this local ancestral genotype. So the question is, what does it kind of represent? And how many do we actually find? So if we look at how many of these sites we find with respect to window size, the first thing to notice is this kind of maybe unexpected rise and then fall in the number as you increase the window size. This is largely an artifact of the fact that small windows have or are less likely to contain SNPs in them. And we exclude windows which don't have SNPs because we don't have enough information to kind of make good judgment about the ancestral inference there. So if you look at fraction of tested sites, you see kind of the more expected picture of the number of sites which have significant population differences dropping as the window size increases. And what that means is that these are not kind of effects of overall admixture, that these effects capture likely variants which are tagged by the local ancestry. So allele frequency differences or even private variants, which are specific to a local ancestry and differentiated to ancestries. So kind of local, like cis ancestral genetic effects. And this is kind of confirmed when we actually bring the real genotypes in and we compare models of DNA methylation based on either the local ancestry or the observed genotypes, which, you know, to some extent capture some ancestral information, because even in observed genotypes, there will be allele frequency differences, but they might not tag kind of unobserved genetic differences between the populations. We find that local ancestry in most cases provides a better model, a significantly better model than the genotype, even if we include the genotype in the model. However, what you might notice is that this fraction is much, much smaller than the 10% that I mentioned at the beginning as a kind of amount of differences you would see between populations in other studies. So one conclusion one could draw is that, okay, only a small fraction of these population differences are driven by genetic effects, and most is environmental. However, there is another problem. While our study had like 130 individuals and the population comparisons were based on a kind of comparable number, so like 60 and 70 individuals, usually around that number. Once you start looking at local ancestry, you are breaking down your 130 individuals into smaller subgroups within these individuals that you're comparing. So if we look across the different methylation sites that we test at the effective number of individuals of pure ancestry that we have in our comparisons, we see that, you know, for the Europeans, we've got usually most around like the kind of 30 to 40 individuals, but for the others, we are down to below 20 individuals in each individual comparison. So the obvious, well, the alternative explanation for why we might see so many fewer population DNA methylation sites with this approach is that we've just got much smaller sample sizes. And so to check that, we took the data from the population comparisons and subsampled it and looked at how many we would expect to find with comparable sample sizes to what we have in each individual comparison. And you can see that if you look at On the right, you see for different sample sizes, the kind of or the number of differently methylated sites in the African to European population comparison. And you can see that when you get down to the sample size of around 20, which is where we will mostly be due to the low number of kind of African and Native American population individuals that we have in each kind of local comparison, the number of heads that you find is comparable to what we see. So the more likely explanation is that actually most of these population differences are driven by genetic effects. And if we had more admixed individuals, we would find more population specific DNA methylation sites. Right. So where are these population DNA methylation sites that we find? So here I'm plotting in dark the kind of observed fraction of the sites depending on their position of the site relative to genes and relative to CPG islands. And the blue categories are essentially depleted, and the red categories are enriched for these population DNA methylation sites. The one disappointing thing, I guess, is that we see depletion around essentially transcription start sites, so promoters, which is actually in contrast to what the studies which compared populations directly found. They reported an enrichment of these population-specific methylation sites at promoters. So there are essentially two possible explanations, one more exciting than the other. The more exciting explanation would be that the population-specific DNA methylation sites which are at promoters are largely environment-driven, and that's why we don't find them in the same fraction, whereas the ones which are not at promoters are largely genetically driven, so we find those, which would be very interesting. The kind of more boring explanation is that there's differential power in identifying differences near promoters versus other genetic contexts, and the only way to find out is to increase sample size. Okay. Then another thing we looked at is because we had DNA methylation assayed in the same individuals across two different tissues. So we did the analysis in both tissues, and we compared whether these population-specific DNA methylation sites are kind of tissue-specific or whether there's large differences between tissue. And at least for blood and colon, it seems that they are largely conserved. So we see essentially very large correlations in effects between, or estimated effects between the two tissues, and also large overlap in the identified DNA methylation sites. So again, suggesting that there's, at least for these tissues, not much tissue specificity for these sites. So to conclude, essentially admixture gives a very nice natural randomized experiment for DNA methylation, but kind of more generally for any kind of phenotype that you can localize in the genome. So any phenotype that you can assign a specific position in the genome. If you look at admixed individuals, you're kind of looking at a very nice, naturally randomized experiment where the local genetic context is randomized with respect to the environment for that phenotype. Obviously, for methylation, it's very important or very relevant because of the environmental effect on methylations, but it's probably also interesting for other kind of genomic phenotypes. In the specific context of the original question, it looks like the majority of population differences between populations and DNA methylations are likely driven by cisgenetics rather than environmental differences. And these effects are likely consistent across tissue. There's some open questions for somebody to look at, in particular this missing enrichment in promoters is, I think, interesting. And for that, we just need larger sample sizes. And I think I'll finish here. And yes, if anybody's, sorry. I shouldn't have said that. If anybody's interested, this work is actually relatively old, from 2017. The paper is online, or you can ask me questions. And obviously, I would like to thank our collaborators, especially in Colombia, and especially those working in hospitals and actually recruiting the patients. Thank you very much.
So that's very interesting there, this difference at promoters.
Yes.
Could it also be that, I mean, most of the genes have alternative TSSs, right?
Yes.
And because you look at admixtures, could it be population-specific TSSs? And if you go in the databases, the default annotation is probably European ancestry TSS. So
I mean, the one thing is we use the same annotations that they used in the population comparisons. So I agree that it's possible that that's the case, but we should have seen the same. We should have seen the same enrichment because we looked at the same things. Yeah. But yes, I agree that it might be another explanation, the differential use of TSS.
Okay, thanks.